Hello and welcome to Nikolai's genetics lessons, and today I have prepared another problem for you about hemoglobins. And here's the problem: the iron atom in the heme group is held in place by direct contact with, and you have to choose one of these answers. Today's question is really interesting because if you'll take a look at the 20 amino acids, you will find that R groups of many have groups which can be reactive, like alcohol group here, or here, for example, like secondary carboxylic acid group here, and for example this amino groups in basic amino acids. But only one amino acid, which is cysteine, can make covalent bond in R group. And this covalent bond cysteine can make not with any other R group of any other amino acid, but with itself, and we call this disulfide bridge, because we see here sulfur. So this information is probably many of you know, but not probably many of you know that another amino acid can make covalent bond in R group. And this amino acid is histidine. Take a look at another picture. On this picture you can see hemoglobin protein, which is here, and here is the heme groups embedded in these proteins, alpha and beta chains. And here in the middle is molecular structure of the heme group, and you see iron here in the middle. And according to this picture, you see that iron is covalently bound to 4 nitrogens, and this is coordinate-covalent bond, because both electrons for such covalent bond comes only from one atom, from nitrogen. But how this heme group is embedded in the hemoglobin? Take a look. This molecule has one part which is hydrophilic. So you see oxygens here and negative charge. And as you know, molecules of water is slightly negatively charged on one side and have positive charge on the other side. So this part is going to be hydrophilic, and we will see it here outside of the protein. And the other part is going to be hydrophobic, and we are going to see it inside, because inside of this globular protein is going to be hydrophobic. But hydrophilic and hydrophobic forces is not enough to hold heme group inside the protein, and actually this plane and all these atoms are in the same plane are held together by histidine. So R group in histidine has a covalent bond with this iron. Take a look at another picture, and here we can see histidine, and with blue color we see nitrogens here, and there are 2 nitrogens in R group of the histidine. And here is a protein chain. And on the picture on the right, you can see these interactions even more clear. This is central alpha carbon, carboxylic group, and here's a protein chain, and here's the amino group and protein chain. And here's the R group. We see one nitrogen, another nitrogen here, and here is a covalent bond to the iron in the heme group. So if you wondered, just like me, if R groups of other amino acid except cysteine can make covalent bond, yes, this is going to be histidine. Now let's return to our question. The iron atom in the heme group is held in place by direct contact with, and now you know that this is going to be nitrogens in heme group, so this nitrogens here, and it's going to be nitrogen in histidine. So this is going to be variant E. And this is all for today. Subscribe and see you in the next video. Goodbye.